So I'm Pat Brown from the Walnut Improvement Program, and we've been around, or the program has been around for over 50 years and has released 23 varieties, most recently Wolfskill, which we released in 2021. And I'm going to just sort of give you an overview of what we've been doing and what we're thinking of next and how you fit into this. So this is who we are. I'm relatively new. I got here 4.5 years ago. Chuck is the elder breeder. He's been here 35 odd years. And then we have 3 young people, Eileen, Stephen, and Christina, taking care of the field and greenhouse, tissue culture lab, and molecular lab, respectively. And I'm going to give my thank yous at the beginning here. Oops. So big thank you to the California Walnut Board for providing funding. We couldn't do anything without you all. And also thanks to the farm advisors and the growers who we work with sort of constantly to test our stuff. So we couldn't do any of this without you. Okay, so what do we actually do? So we spend a lot of our time in the spring crossing walnuts. So here's a picture of the female pistillate flowers right before, right as they're becoming receptive. And the stigmas are barely poking out. So we put bags on these flowers before they're receptive. You can see here's some trees here. Then separately, we'll collect catkins. So here's Chuck with a big load of catkins, and we sort of vibrate the pollen from the catkins through a wire mesh onto butcher paper, and then store the pollen in the freezer. And it stays viable for up to a couple of years at least. Then we'll come back and we'll put the stored pollen into a syringe and apply the pollen, basically poke the syringe through the clear plastic window of these bags to make sure that we get some pollen onto the flowers. So that's basically the process for doing controlled crosses, where we choose the mother and the father of the cross that we wish to make. So after we've made those crosses, we plant out our nuts and we grow them for a while, and then we have to evaluate their performance. And so there's basically 2 main sets of traits that we look at. There are the traits that we measure out in the orchard, including phenology, so leafing date, harvest date, flowering date, also yield scores, blight scoring, stuff like that. Then there are the traits that we measure during crackout, so back over the winter time indoors. And so that's all the nut and kernel quality traits, kernel yield, pellicle color. And here I'm showing how we've recently transitioned to evaluating kernel color or pellicle color from image data. And these are the same trays that Bruce showed earlier, except we spray paint ours blue instead of green. We also spend a considerable amount of time and effort on maintaining germplasm. So we do this both in vitro in the tissue culture lab and in orchards out in the field. And so in the tissue culture lab, we maintain about 1,000 cultures. These are mostly experimental rootstocks. And so in all likelihood, the next rootstock to be released for walnuts will have started out its life as inside one of these tiny little tubes on the first floor of Wixon Hall here on campus. Out in the field, we maintain about 50 acres. That's everything across the breeding program, including a couple of blocks where we maintain germplasm and genetic stocks that aren't maintained elsewhere. Okay, so here's a breeding scheme that gives sort of an overview of our process. So we make controlled crosses every year, generate lots of new nuts. Those get planted into seedling blocks. Our seedling blocks are planted on really close spacing, so 6 feet or 20 feet between rows, but 6 feet within the row. And each individual is unique. It's ungrafted on its own roots. Then we take the very, very best, most promising stuff from the seedling blocks, say less than 1% of individuals, we'll move them into selection blocks, where then now we've got something closer to a commercial spacing, 20 foot by 20 foot. And then these are grafted onto real rootstocks and then we can evaluate them more. The next step after the selection block is to move out to grower trials. So I'm going to return to this at the end, but this is kind of really the crucial step where we think something is good, but then we really have to get it out into the real world before we know. And you all will sort of be the ultimate arbiters of whether something is good or not. The best stuff coming out of grower trials gets released, and then we're typically using a lot of released varieties as parents of the next generation. So one of the things when I arrived that I did first is bring DNA information into this process. And so really the biggest benefit that the DNA information can provide is in this first step here. Instead of planting 100% of our new nuts into the seedling blocks with DNA information, we can throw away some of those nuts before they even get planted. And so that's a big increase in efficiency. And so really what we're trying to do here is reduce our field costs, increase our efficiency by sort of really paring things down, narrowing things down before they even get planted into the field. So that's, I think, the big benefit that DNA information can provide. And then if we kind of look back at the history of the breeding program, our 23 releases so far, this slide is just to sort of show what we've accomplished. So the breeding program was set up in the first place to combine the lateral bearing from Payne with the late leafing from varieties like Franquette and Waterloo. So these are indicated with the blue and the orange respectively. And you can see all of the three most popular varieties we grow today combine both of those things. So the breeding program was successful in doing that. Our most recent release, Wolfskill, is lateral bearing, but it does not carry the late leafing allele. And the same is true for Solano and Ivanhoe as well. And I'm going to get back to that later about why we think that with our sort of climate change and the reductions in chill hours, why we think that this late leafing allele is not really the way to go in the future. So I'll get back to that. But in the meantime, we've released Wolfskill. Here's the tree, here's the nut. Here is the person it was named after, John R. Wolfskill, one of the first settlers in this area. And why you should consider Wolfskill. So it's got here it is sort of lined up against all of the other options that you've got. So Wolfskill, it's going to leaf fairly early, and it's going to harvest sort of in the middle of the season after Vina Solano, but a harvest well before Tulare and Howard. And so really what we think the big selling points for Wolfskill are highlighted here. So if you compare it to Chandler, well, first of all, it looks a lot like Chandler, and in blind testing, a diamond, it's often mistaken for Chandler. Wolfskill kernels are, but it's a little bit larger kernel, a little bit better filled kernel, so higher kernel yield. And at least in the test, in the data we've got so far, we think the color, if anything, is actually a little better than Chandler. So if you've got space and the ability to do so, I definitely encourage you to give Wolfskill a try. Okay, so transitioning now to sort of what the goals of the Walnut Improvement Program are for the future, so where we're at and where we're going. So we're well aware of the fact that this is a challenging time for the walnut industry, and I think the keys to our success really lie in two things. I think we need to increase grower profitability on the one side, and then I think we also have to drive consumer demand by getting people to eat and buy more walnuts. And so there's really like a couple of components to this. We've already developed markers up here in this quadrant for lateral bearing, which also affects precocity and yield, and also for this late leafing allele, which is a mechanism of blight avoidance. Down here in black, though, are the things that I think we need to develop markers for. So we need markers for blight resistance, for chill requirement, for kernel color, and for shelf life, because if someone bites into a rancid walnut, chances are he or she's not going to buy too many more walnuts after that. So Getting back to the markers we've already developed for late leafing and for terminal bearing, this slide is just to show how we can use them in prediction. And so what I'm showing here is a chart showing the average lateral bearing. So this is the percentage of lateral buds bearing nuts in this population of trees that we have in the breeding program. And so you can see there's 9 bars here, and that's because for these 2 genes or regions of the genome that we have markers for, there are 3 genotypes possible at each of them. So basically for lateral bearing, you can be a homozygote for the lateral bearing allele, shown here, or you can be a heterozygote and have 1 lateral bearing allele and 1 terminal bearing allele, or you can have 2 terminal bearing alleles and be a homozygote for that. Similarly, for the early late leafing region of the genome, you can be a homozygote for late leafing, shown here as LL, or the heterozygote or homozygote for early leafing, shown as EE. And so what we're doing is, we're basically, when we use our markers, we throw away anything that is homozygous terminal bearing and anything that is homozygous late leafing. So basically we're saying you need to have at least 1 allele of lateral bearing and 1 allele of early leafing. Otherwise, we're going to throw you out. That nut gets thrown away. The interesting thing is, so now I'm throwing up the names of some of the varieties that are most widely grown. So this is where they fall in this schematic. And you can see that Chandler, Howard, Durham, these are heterozygotes for both of these regions. And in the grand scheme of things, you know, the number of laterals bearing nuts for this combination is not terribly impressive, right? So, you know, Tulare happens to be a homozygote for the lateral bearing allele. Wolfskill, Solano, and Ivanhoe happen to be homozygotes for the early leafing allele. Both of these combinations we expect are going to be better for you all moving forward. The interesting thing is what we predict is going to be the best combination of all, homozygote early leafing and homozygote lateral bearing. We don't have any cultivars at all out there that have this combination. And so this is just an example of how the markers can give us sort of a blueprint for moving forward. You know, not to say that everything in this category is going to yield more than everything in this category, but on average, we predict that it should. And so on average, getting stuff out to all of you from this category here, I think should make a real difference. Okay, so other stuff we've got to do, we've got to develop new, more quantitative assays for everything else. And that's going to enable us to develop predictive markers. So here's our blue trays that we use to collect quantitative color data. At this point, we've got, you know, thousands of images and hundreds of thousands of kernels. And here's just sort of some schematics of comparing the image data with the human scored data, where we score something as a blank or we score it on this 4-point DFA scale. And you can see for the most part, it matches up pretty well. We have picked up on a lot of human errors once we really start to dig in depth into comparing the human and the machine vision. And then perhaps the most interesting thing to me is when we take these pictures, we can take an initial picture right after we crack out the nuts, and then we can leave the trays at room temperature for several months and then take additional pictures over time. And so you can see in the initial picture here on the left, there's a number of columns. Each column is from a different tree. So there's a number of columns in the initial picture that look fairly light. But then after 4 months storage at room temperature, a lot of these have darkened a little bit. And this third tree in the tray really looks pretty interesting because it seems to be able to hold its good color better than the others during storage at room temperature. Okay, another thing we've really got to focus on is chill requirement. So it's no secret that, you know, we could argue about exactly how the climate's going to change over the coming decades, but, you know, chilling hours or portions or whatever you want to call them, they're probably not going to increase. And so if you look at the early data on chill requirement in walnuts, and this is from a growth chamber assay, you can see Chandler's chill requirement is fairly high. So the message here is that we can't rely on Chandler forever. We've got to come up with viable alternatives to Chandler. And, you know, in the breeding program, we've been taking leafing date data for a very long time. I'm showing here a drone picture where you can hopefully see there are some trees that haven't leafed out at all. They're just sort of the bare white skeletons. And then there are some trees down here that are just starting to leaf out, and then some that have leafed out for a while. So we can get really accurate measurements of leafing date. Problem with leafing date is that it's actually, the way I think about it, it's sort of a combination of two things. Leafing date reflects both the chilling requirement and then also how quickly or how many heat units are required for leafing after the chill requirement has been met. And so we feel that a more direct measurement of just chill requirement is going to be with these growth chamber assays, where we cut sticks throughout the winter. So say we go on January 1st, we cut sticks from walnut trees, put them in water in a growth chamber with heat and light, and then we see if the buds ever push on these sticks. And so if they do, you know, if we're cutting these sticks in January and we can get them to push buds in the growth chamber, then that means that that tree's chilling requirement had been met at the time those sticks were cut. And if the buds don't wake up, then it means the chilling requirement hadn't been met, and it's a higher chill variety. So this is now an assay we're trying to develop. Okay, so kind of into the final phase of my presentation, and the one I'm most excited about, where I get to show sort of the data of all the genetic diversity in the breeding program. So what I'm showing to you guys here is a plot where each point represents an individual scion or variety. There are about 1,200 of them. And I'm only showing the ones for which we have at least 3 years of data from the last 5 years. So stuff that we've discarded isn't on here. This is sort of stuff that's still in the running to be the next variety. And so this slide is showing phenology. So on the x-axis, we've got leafing date. On the y-axis, we have harvest date. And you can see there's a pretty high correlation. So earlier leafing means earlier harvest. And then Chandler is highlighted here with the vertical and horizontal dashed lines, right? So Chandler is got fairly late leafing and harvest. And if you look at all of these 1,200 items that we have in the breeding program, most of them fall into this quadrant here on the lower left, meaning that they leaf earlier than Chandler and they harvest earlier than Chandler. So that's been the focus of the breeding program for a long time. So what I'm doing now is layering in our blight data onto those points. And so we give every tree a blight score, 0 to 9, and a low score is good. So anything with a blight score of 2 or less here is a black dot, and that's good. And then as the blight scores get higher, the dots turn orange or red for the very worst stuff. So it's no secret why, at the beginning of the breeding program, the attempt was to combine lateral bearing with late leafing. And that's because late leafing gives blight avoidance. And you can see that if you look at this chart, most of the dots in these 3 quadrants here, leafing and harvesting after Chandler, are black, so they don't get blight. But for chill reasons, I think we definitely need to move towards earlier leafing. This could help give us earlier harvest as well. The worry is about blight. And so the question is, do we have adequate blight resistance within the breeding program? And so just by looking at this picture, I would say, yes, we probably do. There's a lot of dots in here. There's a lot of orange and red, but there's also a lot of black dots in here. And so it appears, given the data we have, that we have some early selections that don't get much blight. But I'm just going to say that the proof is in the pudding. So we're not going to know for sure about any one of these until we get them out in the real world, until they get planted at multiple locations up and down the valley, and until growers like you plant them and figure out for real if if their blight resistance holds up across locations in years. So that's got to be the push for us, is getting this stuff out. Here's another look at the genetic diversity in the breeding program. So what I'm showing now is kernel yield, or what we call percent kernel, along the x-axis, and then shell thickness on the y-axis. And so there's a fairly strong negative correlation here. And so as kernel yield goes up, the shell thickness is going down. I'm getting thinner shells, so that makes sense. And I guess the question here is, well, we'd all like to maximize kernel yield, but can we do this while maintaining shells that are strong enough? And so now in this slide, I'm layering in information on seal strength. And so we rate seal strength also on sort of a 1 to 9 scale. And basically here, any black dot means that the seal strength is okay. It's over 4.5. So we think that anything over that should be okay. As we get weaker, you turn to orange or you turn to red as the seals become progressively more suspect. And so here again, you can, well, actually it's a little bit different here in that we do see selections with fairly weak seals regardless of what the kernel yield or the shell thickness is, which is kind of interesting. But again, if we look at this portion of the graph, so selections with kernel yields over 60%, there are still some in here whose seals appear to be just fine. If we look at these, getting up close to 70% kernel, I'm not sure how much I would trust a shell thickness of 0.8 millimeters, even if the seal appears fine. There's got to be some point at which the shell is just too paper thin. But certainly we have a lot of selections in the 60 to 65% range where I think the shell thickness is adequate and the seal appears to be fine. And so, the question just is, are these seals and shells really strong enough? And again, it goes to the need to get these selections out into the real world, out into grower trials, and because that's really the only way we're going to know for sure whether any one of these individual selections is going to pan out and be the next great variety. So in conclusion, I'm asking for your help, all of you out there who grow walnuts. The Walnut Improvement Program needs you. And so this is how the system works. Basically, if you're interested in growing some of these unreleased selections, we'll work with you to determine what best fits your needs or interests. Say you're putting in a block of Wolfskill or a block of Chandler or Tulare or whatever, and you're willing to try some selections that have similar phenologies to whatever is going in the rest of your block. We'll work with you to get you selections that fit that. Or say you're interested in trying out stuff with really high kernel yield. We'll work together and pick some out that fit your interests. So we will provide you with graftwood, budwood, or new this year, finished trees. You grow them, and then we coordinate with you to come back and collect samples every year, and also data, both from the unreleased selections that we've given you and from nearby reference cultivars. So it's very important for us to have a reference, say, a block of Chandler or whatever it is that's nearby, so we can sort of calibrate the data we're getting from the selections in your orchard. What's in it for you? Well, you get first access to these future releases. And then also we're trying to add some incentives for you to sort of really jumpstart this process. So you can get feedback on how your orchard performs relative to all the other anonymous orchards across the state. So we'll give you back that data. We'll have a recognition process, bragging rights basically for best color as determined by the machine vision for each cultivar. And then finally, you get a few finished trees. So that never hurts. So please, here's my contact info. If you're interested, please get in touch. And thanks again to the Walnut Board. And I think I'll leave it there and take questions.
Thank you. Excellent. So there are two questions from Bob Beatty. Will you be able to see, Pat, the Q&A? Can we access that?
Yes.
Probably it's easier to see that way than me talk. Yeah.
I got it. So Bob Beatty is saying with 63% edible, how susceptible to shell cracking is wolf skill? Let's see, 63% edible. Yeah, Bob, the 63% was the extra lights. So our percent kernel for wolf skill, our 5-year average is 57. So I'm not too worried about wolf skill at 57%. But you know, so the question still goes to, is 63% too high? Is it possible to build a nut with a strong enough seal and shell at 63%? I think maybe it is, but the proof is in the pudding, so I can talk all day. Another question from Bob. Can you describe the growth habit of wolf skill in contrast to existing varieties? Chandler and Sir, great. Ivanhoe only fair. Yeah, Ivanhoe makes more nuts than leaves. Yeah, so I would say that, so even though wolf skill is an early, sort of an earlier leafer, I think its growth habit is pretty similar to Chandler. I don't know if it has quite the vigor of Sir, but it's definitely got a lot more vegetative vigor, bigger tree than Ivanhoe. So I'd say it's not anywhere close to Ivanhoe. Is it as vigorous as Sir? Not sure, but it's vigorous.
I'll go ahead. I'll chime in and ask a question. And maybe you covered this and I missed it, Pat. For the wolf skill, since it is an earlier bloomer, an earlier leafer, what are your thoughts on blight related to wolf skill?
I would spray it if the situation demands.
Thank you. Also, Pat, where does a lot of the funding come from for the Walnut Improvement Program?
So we get funding from the California Walnut Board. So basically the funding comes from you all. We get funding from other sources too, state and federal. But really none of this would be possible without sort of the continuity of funding that we've gotten from the California Walnut Board. So thank you, California Walnut Board.